Hi everyone, welcome to this lecture for chapter 6. So in chapter 6 we are talking about proteins. So this will be the third of our 3 macronutrients that we learn about. Alrighty. So proteins, again, we'll review some of the same things we've said before. So proteins are one of our 3 macronutrients. Of course, by now, hopefully you can recall that carbohydrates and lipids or fat, those are 2 other macronutrients. So macronutrients, remember by definition, these are large molecules that are made up of smaller molecules. So in the case of proteins, these amino acids are the smaller molecules that make up proteins. So we could say that amino acids are the building blocks of proteins. Remember, another defining characteristic of macronutrient is that it provides energy or calories. So proteins do provide calories to the body. Of the 3, carbohydrate, fat, and protein, protein is the one we will use the least often for actual calorie or fuel requirements. Primarily, we're going to use carbohydrate and we're going to use fat for fuel, but do understand that proteins do contain calories as well. The reason they contain calories is because of these same 3 elements, carbon, hydrogen, and oxygen, that we see in carbohydrates and fats. So that's yet a third defining characteristic of macronutrients. All 3 of these, carbohydrates, fats, and proteins, they all contain carbon, hydrogen, and oxygen. You'll remember that, you might remember that carbohydrates, it's C6H12O6. Remember, the glucose is a hexagonal molecule. The fat, remember it was these, we have the glycerol backbone, which is carbons, and then we have these fatty acid chains, which are also carbons with hydrogens bound to them. And then you might remember these carbons right here that attach to the glycerol backbone also are bound to an oxygen and an alcohol group. So remember, fat and carbohydrate, same building blocks, carbon, hydrogen, and oxygen. A new piece here with proteins is that proteins contain nitrogen. So proteins contain carbon, hydrogen, and oxygen, and they contain nitrogen. So that's new. You definitely need to understand that that's a differential of proteins relative to carbohydrates and fats. So another important thing about proteins that is different is that we find protein in all of the tissues in our bodies, and we would find them in the bodies, I guess, of other living things as well, animals and plants too. Proteins are also these complex molecules that we assemble or make ourselves. And so this line here that proteins are dictated by genetic material or our DNA, what that means is that our DNA quite literally codes for the proteins that our bodies will make at any given time. So whereas with like fat or carbohydrate, we basically utilize what we get from our food. With proteins, it's a very different story. When we ingest proteins, when we eat proteins from our food, we break them down to their amino acids, but we don't use the amino acids in the same way that we use glucose or fructose, or the same way that we use the fatty acid chains or cholesterol backbone. Rather, with amino acids, we reassemble them into our own body proteins, and then they become these structural pieces of our bodies. So whereas with carbohydrate and fat, we primarily use those for fuel, with proteins, very different story. We disassemble the proteins that we eat to harness the amino acids, and then we reassemble those amino acids into the structural components of our bodies. So with amino acids, definitely familiarize yourself with this term. hopefully by the end of this lecture and by the end of this class, really, amino acids will be as fluent of a word for you as protein itself. So amino acids are the building blocks of proteins. We can categorize them by essential amino acids and non-essential amino acids. Remember, we saw this term essential already, right? We saw essential fatty acids. And what did that mean for a fatty acid to be essential? It meant that it must be eaten. We must obtain it from our food, right? So essential amino acids, we must eat them in our food. And this is because we can't produce them by ourselves. We can't produce them in our own bodies. And then the non-essential amino acids, these are amino acids that can be synthesized by the human body. So they are non-essential to our diet. We don't have to eat them because we can make them from one another, actually. So we can take some amino acids and convert them into other amino acids. And that's what our non-essential amino acids are, the ones that we can make. So let's take a look at the amino acid structure. So again, an important thing to remember here, what I would encourage you to do actually, like in your studying, would be to draw out the structures of the building blocks of carbohydrate, draw out the structures of the building blocks of triglycerides, of phospholipids and sterols, and then draw out this structure of amino acid, and then even draw out the structure of a protein. But you'll see on the coming slides, the complete structure of a protein is not actually very drawable. It's pretty hard to draw because they're such complex molecules. So do know the amino acid structure at the very least. And we have over here 3 types of amino acids: glycine, leucine, and aspartic acid, or aspartate, you might see. And so you'll notice they all have the same structure, right? So that structure involves a central carbon, right? It's all highlighted here, right? The central carbon, right in the middle, and you see it has 4 binding sites, just like we've learned when we talked about saturated, unsaturated fats. So it has 4 binding sites. All amino acids are going to have a central carbon that's attached to 1 lone hydrogen. Then it's also going to be attached to what we call the amine group, which is a nitrogen and 2 hydrogens. So over here in example A, here's your amine group, highlighted in pink or purple. And then they're all going to have this acid group, which, remember, we saw this with our fatty acid chains. This is called a carboxyl group, where you have a carbon bound to an oxygen and an alcohol group. So it's called a carboxyl group. This is a carboxyl. And then what's different is this side chain. So the side chain, let me highlight this for you. The side chain varies for each amino acid. So this is how we get close to 20 different amino acids. So let's take a look here at the side chain for glycine. It's just another hydrogen. Isn't that interesting? The side chain for leucine, we have 4 additional carbons with their associated hydrogens. And the side chain for aspartic acid is another carbon with actually another carboxyl group. Huh, interesting. So the side chain is really important because that's what makes each different amino acid. So on down here in the one that I'm drawing, I'm just going to say usually the side chain is abbreviated as the letter R. So R equals side chain. R is the shorthand way to say side chain. So do understand basic amino acid structure. This, I'm not going to test you on the individual amino acids. I'm going to be honest with you, I don't even have them all memorized myself. But if you're interested, you can take a look at the essential amino acids and the non-essential amino acids. All that I want you to do is be able to define non-essential and define essential. So know what differentiates the two. But if you're interested, the essential amino acids, the one we must consume in our food: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. And then the non-essential amino acids, these we can make. We have alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. So as you go through learning about nutrition, even beyond this class, you might come across any of these words. So hopefully you'll start to be able to recognize some of them as amino acids. You'll notice not all of them, but a common theme is that they end in this ene, I-N-E, which is related to, again, that amine group, which is that nitrogen group. So let me point that out too, because I didn't point that out when I was on this slide just a moment ago. Notice that in the side chains, we're dealing with carbon, hydrogen, and oxygen. Notice that in this part of the backbone, we're dealing with carbon, hydrogen, and oxygen, right? Just as we've said, carbon, hydrogen, and oxygen. Our fats, our carbohydrates, and our proteins are all made up of carbon, hydrogen, and oxygen. And then we said that amino acids or proteins are unique in that they have a nitrogen in them. We will see that some amino acids might also have a sulfur, which is abbreviated F. They might also have a sulfur atom in their side chains, but not all of them. But all amino acids have this amine group, hence being called amino acids, because they have the amine group and they have the carboxyl or acid group. So that's why they're called amino acids. So you'll see that most of these amino acids end in this INE name, although not all of them, like tryptophan. Okay, so amino acid is just the starting place, right? So we have to get from amino acid to a full protein. So here we're looking at a carbohydrate, a complex carbohydrate, which, for example, would be starch or fiber. And then letter B is showing us an example of a protein, such as insulin. Insulin is a protein. It's a hormone. It's a protein. It's a hormone that is a protein. So let's take a look here. You'll start to notice each of these amino acids has a 3-letter abbreviation. This is alanine, lysine, proline, threonine, tyrosine, phenylalanine, glycine, arginine. You don't need to, again, know the abbreviations, but even just a quick look back here, they're pretty easy to guess because it just starts with the first 3 letters of the name for the most part. So where building the full macronutrient is pretty similar with proteins and carbohydrates. So carbohydrates are just these long chains of glucose molecules. And same thing, proteins are just these long chains of amino acids. Where they start to differ is that proteins, are actually 2 chains bound to each other. So do you see how right here and here, we actually have a second chain that gets bound to the first chain, which I'll draw through in blue. So you see how we have 2 chains there? And you'll see they are bound at what we call disulfide bridges. And so these are sulfur compounds. So these sulfurs, and cysteine is an amino acid that has sulfur in its side chain. So you'll see where there's cysteine, we can bind these 2 amino acid chains to each other. Same thing here. And then you'll also notice that the chain that's in red, you'll notice that even 2 of its own cysteine molecules are bound, its own cysteine amino acids are bound to each other. So it starts to fold this amino acid chain on itself. So these are 2 important pieces to understand about proteins. This is why I say they are actually quite complex molecules, because it's 2 amino acid chains bound to each other, and each chain may be folded back on itself. They aren't always, as you can see, the blue one isn't folded on itself, but a lot of times there will be folding. And it's this level of specificity and individuality in the amino acid chains that dictates which a protein it is, right? So that we know that we're building the protein insulin in this case. Alrighty, so we are going to talk more about this in a few slides, but I guess I don't like the order of these slides. We're now going to talk again about this process of building the non-essential amino acids. So skip over this. That's this process of transamination. So transamination is basically how we produce the non-essential amino acids in our bodies. And so really all we're doing is transferring the amine group from an essential amino acid to a different carboxyl group with a different side chain. So that's all we do. So right here, this is the schematic of transamination. So we have this, right? This is kind of almost the backbone for an amino acid, right? But what's missing is the amine group, and what's missing is the specific side chain. Oh, I'm sorry. This is the side chain right here. Yeah, what we're missing is just the random standalone hydrogen. So this is the side chain. Remember glycine we saw in the beginning, glycine's side chain is just the hydrogen. So all we're doing here is we have this amino acid backbone, but it's actually missing the amino group. So valine is going to donate its amino group, its amine group. So it gets transferred over here. You can see if I erase my crazy lines. So the amine group gets transferred over here to this, what would be glycine backbone. And now we actually have glycine now that the amine group is attached. So that's all it is transferring the amine group, usually from an essential amino acid. So remember, this is valine here. And if we go back and look on our chart, the essential amino acids, boom, there's valine. So usually we can build these non-essential amino acids from the essential amino acids, right? Sometimes we can build them from other non-essential amino acids, but oftentimes we'll start with an essential amino acid, and then we transaminate, right? We move the amine group. So I will test you on this word transamination. Again, you don't have to memorize it because it already tells you what it is. It's transferring the amine group to build our non-essential amino acids. We do also have what are called conditionally essential amino acids, and these are times when non-essential amino acids become essential. I won't ask you to remember this concept, but for example, we will talk about this condition at the end called phenylketonuria, in which the normally non-essential amino acid tyrosine becomes essential. So in this case of phenylketonuria, there's like a genetic issue in how that person's body makes phenylalanine. And so generally they're not making the amino acid phenylalanine very well. And so tyrosine becomes essential so that it can help kind of in place of phenylalanine. So if you go back here, tyrosine is non-essential, phenylalanine is essential. And so when someone has this condition called PKU, phenylketonuria, they're not using phenylalanine exactly correctly. So tyrosine becomes essential in phenylalanine's place. So conditionally essential amino acids, we have some circumstances where non-essential amino acids become essential. Transamination. Okay, so now we'll talk in more depth about how proteins are made. So proteins are made of amino acid chains, and these from amino acid chains, we could call these peptides, peptide chains. So remember we talked about pepsin as one of the digestive enzymes in the stomach, and pepsin digests proteins. So where you start to see this pep, start to associate that with, or peptide, start to associate that with proteins. So peptide or peptide chains is another way to say amino acid chains. And these amino acids are bound together by peptide bonds. So if I go, let me go back to this picture. So these bonds between the amino acids are called peptide bonds. You might remember we have bonds between glucose molecules that are called alpha bonds or beta bonds, right? So same thing, they just have names. The bonds have names, either peptide bonds. And peptide bonds build peptide chains. So we start to call these chains dipeptides, tripeptides, oligopeptides, and polypeptides. Now you've seen those words before too, or at least those prefixes, right? Di, tri, oligo, and poly. So they mean the same thing here. They mean 2, 3, a few, and many, right? So a dipeptide is 2 amino acids, a tripeptide is 3 amino acids, an oligopeptide is like 4 to 9 amino acids, and a polypeptide is like 10 or more, up to hundreds. So it's the same thing. We just build these chains, these polypeptide chains of amino acids. So genetic expression is how we build proteins. So gene expression is the process by which cells use genes to build proteins. Remember back on the first page of this lecture, I said that we make proteins in our bodies. So yes, we eat proteins, but we don't just like use the proteins that we eat. We don't eat beans and use bean proteins. We don't eat cow flesh and use cow proteins. What we do is we break the cow's proteins down, or we break the beans proteins down to their component amino acids. So they're a cow's muscle tissue or a bean's flesh is made up of the same amino acids that we use in our bodies, but the proteins that they make are very different. So we're not going to use their proteins, but we will use the amino acids. And so we have to then build our own proteins, and that's this process of gene expression and you might recall transcription and translation. I don't know why that drew a triangle. It didn't draw a triangle. So know these words: genetic expression, transcription, and translation. So for some reason I have a bunch of other slides in here, which is annoying. Sorry about that. I don't like the order of these slides. So a couple other things here. Gene, right? This is just a segment of your DNA or a section of your DNA that serves as the template for the synthesis of a particular protein. So when we say gene expression, or when we say a gene codes for, this is what we mean. There's a segment of the DNA, which we call a gene, and that gene has the instructions to build or express a particular protein. This word expression is used kind of synonymously with the idea of building a protein. And then nucleotides are the little building blocks of your DNA, and so it's quite literally the sequence of nucleotides that codes for a gene or that codes for a protein. So this is building the this is why these things are out of, well, I guess it's not really out of order, it just feels confusing in my brain. So this is building the polypeptide chain. You have 1 amino acid, 2 amino acids, right? They bind to each other. We actually have dehydration synthesis, so we release water to form this peptide bond. And then we have a dipeptide when we have 1 amino acid bound to a second amino acid, right? 1, 2, dipeptide. Okay, so that picture should kind of come later, to be honest. So let's talk about gene expression, transcription, and translation from the beginning. So here's an example of your DNA, right? You've got the double helix, right? And then these, the G, the A, the T, and the C, these are your nucleotide bases. If you want to give yourself a little quiz, you can remind yourself what they each stand for. We don't need to know that for this class, so I won't bother here. And so literally it's just reading sequences of these nucleotides that will tell us a particular protein to build. And so that's the nucleotide sequence is the gene that codes for a protein. So we have two processes to genetic expression. We have transcription and we have translation. In transcription, we build messenger RNA, which is literally a copy of the genetic code in the DNA. So transcribe, also just think of what this word means. If you transcribe something, what do? Right? Like if you're listening to something and then you write it down word for word, you're transcribing it. You're transcribing something from oral to written. So that's all we're doing here. We're transcribing something from DNA to RNA, and we do that using this messenger RNA. And then we translate it. So first we transcribe it from DNA form to RNA form, and then we translate it into protein form, right? So transcription comes before translation. Hopefully that will make sense just as it is. Again, just using these words as definition. If you want another way to remember it, it's kind of goofy, but transcribe, it's the same letters up to the C or the L, and C comes before L in the alphabet. So transcription before translation. And then again, translation is you're translating it to a whole new language now. So we're translating it from the language of mRNA to the language of protein, right? Translation takes place at the ribosome. For our class, we don't go into very great detail with transcription and translation, which is maybe you're making a giant sigh of relief there. But I do want you to get the basics of it, right? So transcription is happening inside the nucleus, where the DNA, again, is this double helix is going to separate. You're going to reveal a gene, a sequence of nucleotides, and then mRNA. That's hard to see here. The mRNA is this piece here. Starts to copy that genetic code, right? So this is mRNA, and you can see it coming out of the nucleus, mRNA. And then in the cytoplasm is where translation occurs. In the cytoplasm, specifically at the ribosome. So where's the ribosome? These little guys are ribosomes, right, in the cytoplasm. So the mRNA is going to come through the ribosome. The ribosome is basically going to read, right, and translate. So the ribosome is going to read what the mRNA says, and what the mRNA says is it gives us a sequence of amino acids. So the code on the mRNA, I'm here at number 3. The code on the mRNA is translated into the instructions for a specific order of amino acids. Boom. That's translation. So what's going to happen is we have these little transfer RNA, which is abbreviated tRNA. Transfer, or call it transport, but it's transfer RNA, tRNA, is going to be going out in the cytoplasm, finding the specific amino acids that will meet the three nucleotide code on the mRNA. So tRNA is going to bring, you'll see these 3 amino acids, 1, 2, and 3, and they match this 3-digit code on the mRNA. And so that's how it goes. So the tRNA, here's the next one, or that one already went. Here's the next one coming in with its 3 amino acid sequences. So the transfer RNA binds with the specific amino acids in the cytoplasm and then transfers the amino acids to the ribosome based on the code or the specific order dictated by the mRNA. And then in that way, we start to build this amino acid chain. So it's not really shown until down here. You're starting to build this amino acid chain. So this looks like a worm, but each of these little segments on the polypeptide are the amino acids. And so eventually we've built the entire chain. But this is actually only step 1 of DNA synthesis, right? So now you've built this full chain. once the synthesis is complete, the protein is released from the ribosome. The protein may go through further modifications in the cell, or sometimes it can be functional in its current state. So, okay, again, I'll have to reorder these, I apologize. So here would be the next steps in building the protein. So we have the primary structure, which is not written here, but the primary structure is just the amino acid chain. I'll say this is primary. That's how you abbreviate primary, with a little degree sign. That's the primary structure. And then the secondary structure, we start to see these secondary structure down here. You'll start to see these folds, right? Or kind of some bends. So we would see those disulfide bridges again, where certain amino acids kind of bind to each other in the chain, and you start to get bends or folds. And then in the tertiary structure, you basically have like another level of bending and folding. And so you start to get more of a globular compound now, because it starts to become very three-dimensional. And then in quaternary, you have two tertiary polypeptide chains bound to each other. And it's often at the quaternary structure that we see functional, I'm going to abbreviate, proteins, right? And this is what we mean when we say they're so specific, is because first we have to build these complex amino acid chains based on our DNA, and then the chains go through further folding, and then they bind to a whole nother polypeptide chain. So they become very, very, very specific. And that's how we know when we have, for example, insulin versus salivary amylase, right? We don't want insulin in our mouths produced by our salivary glands. We want salivary amylase produced by our salivary glands. And we want the pancreas to make insulin. We don't want the pancreas to make salivary amylase, right? So all of that is based on our genetic information. Okay, so going back a little bit, how are proteins made? So there's this concept of protein turnover, right? Which contributes to the amino acid pool. So right in here, when I say that this transfer RNA can just go out and pick up amino acids free floating in the cytoplasm, that's what we mean by amino acid pool, is that most cells are going to have this very small pool of amino acids that come from our food or from breakdown of certain proteins inside the cell once they're no longer needed. So any cells are always going to have this small pool of amino acid so that the cell can continually build new proteins as needed. So protein turnover, existing proteins are degraded to contribute to the amino acid pool to provide the building blocks for new proteins. So this meets the constantly changing demands and physiological functions of the cell. Just going to erase this because we want you to understand the amino acid pool and the concept of protein turnover. This is a helpful chart too, to kind of show you how we build the amino acid pool. Basically, again, from proteins, amino acids from proteins from our foods, or amino acids from the breakdown of cells or other proteinous structures within the cell. And so then those amino acids are available for us to build non-protein compounds that might require nitrogen. Examples would be serotonin and creatinine, or to build body proteins such as enzymes, antibodies, some hormones like insulin, and various other components of the cell, lipoproteins. Or we would use amino acids to synthesize fat from that carbon, hydrogen, oxygen skeleton, and then we can store that as adipose tissue. So this is kind of an important one to make note of, because I think sometimes in the United States we act like protein is just this endlessly benevolent, amazing compound, which it is. It's very important. I don't mean to discount how important it is. But if we have too much protein, there is such a thing as eating too much protein. And if we consume too much protein, remember the building block of protein are these amino acids that are made up of carbon, hydrogen, and oxygen. So if we have too much amino acid because we ate too much protein, we're going to convert that carbon backbone into triglyceride, and we're going to store it as adipose. Again, remember that process of gluconeogenesis. If we have excessively low blood glucose, if we have a bunch of extra amino acid, we can, or even if we don't have extra amino acid, we can break down our body proteins to use the amino acid carbon skeleton to make glucose that can then be used for energy. And then we will see actually in the next chapter, chapter 7, that some amino acids can go directly into our energy production pathway, the Krebs cycle. Maybe you remember that name, the tricarboxylic acid cycle. Some amino acids can go directly into that process to contribute to energy production. There's no specific reason why I've changed my color here. I only changed my color to highlight this. So that looks a little confusing now that I look back on it. This is also important though, so maybe I'll change my color for that purpose. Make note of these three. So when we're using amino acids, this is energy storage. This is energy in the form of glucose. This is energy directly from the amino acids. When we use amino acids for energy, we have to get rid of that nitrogen, right? Because there's no nitrogen in glucose. There's no nitrogen in triglyceride. So we don't use nitrogen in the energy in harnessing the energy from our food or using the calories from our macronutrients. So that nitrogen we have to get rid of it. So the nitrogen from the amine group, right, from the NH2, gets converted into something called ammonia, which is NH3, and then converted into something called urea at the liver. And then the liver transports urea to the kidneys to be excreted. So there can be harm over a long term if a person continually has too much protein coming into their body, such that they're doing these three processes of converting the amino acid skeleton to triglyceride, or converting it to glucose, or using it for energy. If there's too much of this ammonia urea creation, we can see liver damage and kidney damage in the long run. But again, that's something that would occur over generally years, if not decades. How are proteins made? Again, so the primary structure, secondary, tertiary, and quaternary. Again, I kind of showed you that here. So primary structure is just the amino acid chain in their specific order. Secondary structure, we start to see the twists in the amino acid chain. Tertiary structure, we start to see a 3D shape, and more of those disulfide bridges. And it's really at this point that we start to determine the function of the protein. But then the quaternary structure, we see that with some specific proteins. So remember, not all proteins are going to build to the quaternary stage. Some might even be useful just in the primary structure, but most are going to be useful in the tertiary structure and the quaternary structure. So in the quaternary, we have polypeptides that bind to each other to form larger proteins. And actually, sometimes we can see more than 2 polypeptides bound to each other. Sometimes we can see 3 or 4. And so this is where we start to get more globular or fibrous proteins, like your red blood cells, for example, are protein molecules at the quaternary structure. So protein, again, kind of like in fatty acid chains, we said that the shape of the fatty acid chain determines the liquid or solid in foods. So same thing here, this complete protein structure and organization determines the function of the protein, right? Determines whether this protein is insulin, whether this protein is a red blood cell, whether this protein is a white blood cell, whether this protein is salivary amylase or pepsin, right? So this is really important that you understand that the structure of the protein determines which protein it is and therefore what it does in the body. Red blood cells made up of proteins. So these are globular. This is at that quaternary globular shape, right? Your red blood cells, for example. Okay, so now we also need to understand this term denaturation. Let me just go back to highlighter. Denaturation. So denature, again, just look at what the word means. We're going to denature something. What could that possibly mean, to denature it? We're going to take away its nature, right? We're going to take away its inherent nature. So if we denature a protein, it loses its function. Protein function is lost. It's denatured. It's no longer a functional protein. So how does this happen? Basically, when this complex structure breaks down. So when proteins uncoil, they lose their shape and they lose their function, right? So anytime anything that's going to affect this very delicate quaternary structure and ruin that structure, you're going to ruin or lose the function of the protein. So this happens when the protein is exposed to heat, acid, an acidic environment, a basic environment, heavy metals, or alcohol. We already saw one of these ideas somewhere, right? This idea that proteins would lose their function in an acidic environment. Where did we see that? In the stomach, right? Let me just draw it here, I guess, right? The stomach, we said the stomach has hydrochloric acid, pH of like 2, right? HCl, pH 2. What lost their function there? What lost its function in that superacidic stomach? Your salivary amylases, right? Your lingual lipases, any of the digestive enzymes from your mouth were suddenly denatured because those enzymes are proteins. They're suddenly denatured when they entered the stomach. That's why we didn't see very much carbohydrate digestion or very much fat digestion in the stomach. So how else can this happen during digestion for protein breakdown? Right? I guess we also saw acid in the stomach to denature proteins. Again, if we're eating a black bean, we don't want black bean proteins. We just want the amino acids from the black bean protein. If we're eating cow flesh or pig flesh, we don't want pig proteins. We're not pigs. We're not cows. But we do want the amino acids from their proteins. So we want those amino acids so we can build our own human proteins. So we denature the proteins that exist in our food. Again, denatured enzymes are not functional. If your blood pH gets out of the normal range too, remember we said blood pH is like in the low 7s, kind of like a 7.35, 7.34. If the blood pH gets out of normal range, we can start to actually denature the red blood cells themselves. Also, when we see high fever, like if someone has an infection or an illness, like a more bacterial infection, a viral infection, usually, oftentimes, not always, there might be high fever. And the point of that would be to denature the enzymes from the bacteria or virus. So proteins in the diet. All essential acids must be available to the cells. Okay, well, what do I want to say here? There's this concept of the limiting amino acid. I won't really test you on this, but when we eat protein-rich foods, we eat them for the amino acids. Remember, there's about 20 some odd amino acids. We say 20 in this class, but there's close to 26, to my knowledge, that have been identified. So there's about 20 amino acids. So we need all of them in specific proportions to one another so that we would be able to build any of the proteins in our body that we might need at any given time. So whatever amino acid is in the smallest supply from the diet, we call that the limiting amino acid. And so that limiting amino acid is going to slow down the process of protein synthesis. So limiting amino acids can typically come about from inadequate protein intake. Generally, I guess you might see this more commonly in somebody who might follow a plant-based diet. because some plant foods are limited in certain amino acids, whereas animal flesh or eggs or milk actually generally has a pretty good supply of all of the essential amino acids. So there's this concept of the limiting amino acids. So whichever one is in the smallest supply is going to cause a slowdown in protein synthesis. And that can lead to issues, certainly, like lysine is one of the most common examples that I see, where lysine, which is one of our essential amino acids, like vegetables aren't particularly rich in lysine, nuts aren't particularly rich in lysine, but beans are. And then, of course, like eggs are a good source of lysine, meat can be a good source of lysine. So somebody who might be eating eggs or meat very infrequently, or again, somebody following a vegetarian or vegan diet, or specifically vegan, I guess, or whole food plant-based, if they're not doing their diligence and eating beans or lentils roughly every day, they might become deficient in lysine. And lysine can be an important component of building some of our immune system proteins. So like cold sores, interestingly, are one thing that come about pretty often from lysine deficiencies. So kind of like a benign aspect of like a slightly compromised immune system. Okay, so in the same vein, there's this concept of complete and incomplete proteins. So a complete protein is something that provides all 9 essential amino acids in sufficient amounts. And then incomplete proteins, this is what we find in some, not all of our plant foods, where all of the essential amino acids are not there in proper amounts. So with complete proteins, we see that pretty much all animal sources of protein, again, this would be meat, dairy, and eggs, do provide all 9 essential amino acids in sufficient amounts. In the plant world, we also see that soybeans, quinoa, and some other, I don't keep this list in my head, I want to say potentially hemp seeds or chia seeds, but definitely soybeans and quinoa are also complete proteins. So these plant-rich, and these protein-rich plant foods, soy and quinoa, also provide all 9 essential amino acids in sufficient amounts. With incomplete proteins, I think I put the chart in here. If not, your book has a really cool chart for how to pair incomplete proteins if you are following a vegetarian or vegan diet, so that you can make sure you're getting the proper amount of all 9 essential amino acids. So that's this concept of complementary proteins, where you combine 2 or more foods so that you do supply all 9 essential amino acids. So it's food combining, basically. Mutual supplementation, combine 2 or more incomplete protein sources to make a complete protein. We don't really talk about this. So focus more here on complementary proteins. Yeah, so this is a great chart. I highly recommend this for anyone, especially if you're going to go into the field of nutrition. It doesn't matter if you follow a plant-based diet or not, you're going to encounter a lot of people who do. So being able to help someone understand how to combine protein-rich plant foods that might be limited in a specific amino acid, so that they can have a complete protein meal. So this is super cool. So beans are limited in methionine and cysteine, the essential amino acids methionine and cysteine, but grains, nuts, and seeds are great sources of methionine and cysteine. So build meals that provide legumes with grains, nuts, and seeds, or with grains or with nuts and seeds. So an example would be like hummus, which is chickpeas with sesame seeds, right? So that's going to give you seeds and beans. So hummus theoretically would be a complete protein food. Or classic just beans and rice, you're getting beans, which is a legume, and rice, which is a grain. And then you can go on down and see, like I said, grains are limited in lysine, nuts and seeds, and vegetables. They're all limited in lysine, right? It's an important one for your immune system. So you'll see that with grains, vegetables, nuts and seeds, legumes are going to be an important complementary. So that's why I say legumes, especially someone following a plant-based diet, I do recommend legumes every day. And then of course you do need to also have whole grains, vegetables, nuts, and seeds, especially grains, nuts, and seeds, to be providing the methionine and the methionine and cysteine, because you'll notice the vegetables are also low in methionine and cysteine. So I won't go through this whole page, but again, this is a great chart. Okay, so just like with fat and carbohydrate, let's talk about protein digestion. So remember, we said that carbohydrate digestion begins in the mouth. There's also a small amount of fat digestion that begins in the mouth, but there's really no protein digestion in the mouth. So it's really not until the stomach, when we can denature the proteins, that we see the beginning of protein digestion. Remember, the hydrochloric acid also activates pepsin from the precursor pepsinogen. Pepsin begins the breakdown of proteins into the short polypeptide chains and starts to slough off some individual amino acids. And then remember, gastrin is another digestive hormone that controls the release of hydrochloric acid, as well as pepsinogen. Digestion continues in the small intestine, as is the case with everything. In the small intestine, we see pancreatic proteases. Again, that should be pretty straightforward. You have prote for protein, and aces for enzymes. And we did talk about briefly trypsin, chymotrypsin, and carboxypeptidase. I said you didn't need to know them in chapter 3 when we talked about digestion overview. You still don't necessarily need to know much about them, but for unit 2, at least start to identify that trypsin, chymotrypsin, and carboxypeptidase are proteases, are pancreatic proteases, meaning they are protein digesting enzymes made in the pancreas, secreted into the small intestine. And then, of course, we're going to see some absorption into the enterocytes of the small intestine, absorbing amino acids and small peptide chains, so dipeptides and tripeptides. High doses of individual amino acid supplements can lead to an amino acid toxicity, as well as potential deficiencies of other amino acids. So I don't really recommend dosing, supplementing with amino acids. Of course, that's something to talk to anyone's primary care provider about, but there's plenty of amino acid in our food. It's just a matter of eating enough and eating a good variety of protein-rich foods. So the overview, right, I guess technically we could say that anything is digested in the mouth because of the chewing, so the mechanical digestion. But really it's the stomach where proteins get denatured by hydrochloric acid. So that's what this is showing. Here's your globular protein that suddenly gets broken down into, its peptide chains. So it becomes inactive. And that would be the case, like if you're eating a bloody piece of steak, you don't really want cow's blood. You don't need cow's blood, but you want the amino acids. So you want to break down the cow's red blood cells so that you can render the amino acids from it. And then pepsin is activated to break proteins into single amino acids and smaller peptides, right? Smaller peptides. And then in the small intestine, the pancreas is going to produce and secrete proteases, trypsin, chymotrypsin, and carboxypeptidase. And those proteases digest the polypeptides into smaller amino acids, dipeptides, and tripeptides, which get absorbed into the enterocyte, where dipeptides and tripeptides will get finally broken down into the amino acids, and it is only individual amino acids that get absorbed into the bloodstream. And then the liver, so from the blood, these amino acids are going to go to the liver. And again, they might get converted to glucose or fat if they are in excess. They might get used for energy if the person is in dire need for energy, or we're going to build new proteins or get sent out to various body cells to restock the amino acid pool in those body cells. We're not really going to talk about protein quality or score, but there is this concept of protein quality, which is again based on the limiting amino acids in a food. But So again, animal protein and many soy foods made from soy are going to get a high score because they don't have per se a limiting amino acid. They have a very good amount of all 9 essential amino acids, all of which get absorbed quite well. Your book goes into a little more depth here, and there might even be a question on this at the end of the chapter in the chapter review questions, but I'm not going to test you on this. So feel free to go into this if you're interested, but this is as much as I'll say here. Okay, so I've said now probably a bunch of times in different lectures that proteins are like structural and functional to the human body. So really this idea that they are a source of energy, it's like last resort, right? That's the last thing we want to do with proteins. So here is a short list of various structural and functional purposes of protein in the body. So many of our hormones and enzymes are made from proteins. Right? We've just looked at pepsin, we've just looked at insulin, any of the enzymes we've talked about so far, like salivary, amylase, all that trypsin, the chymotrypsin, the carboxypeptidase, those are all protein enzymes. We'll see coming up that proteins help maintain fluid and electrolyte balance, as well as acid-base balance in the body, or they can be a method of helping with acid-base balance. I've just said that our red blood cells and not many of our white blood cells and immune cells are proteins. So yeah, I skipped over cell growth, repair, and maintenance. Again, this kind of speaks to some of the structural components of our cells, like especially our muscle cells. And even in cell membranes, we have a lot of transport proteins that exist in our cell membranes in that phospholipid bilayer. So growth, repair, and maintenance is all going to be very dependent on those proteins that exist in those phospholipid bilayers. We also talked about lipoproteins that can transport fats, right, that make up our high density, low density lipoproteins, and that help make up our chylomicrons after fat digestion. And also how we store some compounds, some vitamins or minerals or other nutrients the storage can be assisted by proteins. And then also our blood clotting, wound healing, which is again kind of an immune function, and our nervous system all rely on proteins. So this is just a short list, kind of a general overview, but proteins are super important to how the body works, not necessarily as an energy source. So this diagram is just looking at how proteins function in fluid balance. So up top, we're looking at normal fluid balance. And basically what we see is that fluid is going to follow the proteins. So as blood flows through your body, blood pressure is constantly changing throughout the body depending on where there's blood flow. So fluid is going to leave the capillaries and go into like an extracellular space. And then so long as there's enough protein in the blood, fluid is also going to flow back out of that extracellular space and go back into the capillary to be recirculated back to the heart. So we're always going to see this exchange of fluid out of the capillaries into extracellular space, ultimately into the cells if the cells need fluid. But then that fluid should again flow back out, back into the blood, back to the heart, so that we're never like swelling like down here in this case of edema. So this would be normal fluid balance up top. In edema can be caused in some cases by insufficient protein in the bloodstream. So right here, where the fluid would normally flow back into the capillaries because it's following the proteins, if there's not enough protein in the blood, then the fluid is not going to return to the capillaries, so it's going to get stuck here in this extracellular space, and so you get this swelling, literally just pooling of fluid from the cardiovascular system because there isn't enough protein in the blood to pull that fluid back out. So that's one example of the function of proteins in the body, is to support fluid balance. And then I said that we have transport proteins in our phospholipid bilayers in our cell membranes. And we looked at this already. like when we talked about absorption of some nutrients, we talked about how some nutrients require like glucose, for example, requires a transporter, a glucose transporter, to allow glucose to come into the cell. We also see sodium and potassium, two of our key electrolytes, also have transport proteins that allow these minerals to come into or to leave the cell. And again, that can support nerve impulse transmission, that can support muscular contraction. So having these transport proteins is critical to how our bodies work. Okay, so deamination, this is again another important process that I want you to know. And again, just look at the word de, taking away, amination. So we're taking away the amine group, right? The process, the ation of deamination. So this is what happens when, I would go back, but it's several slides back. This is what happens when we have amino acids that we use to make glucose in gluconeogenesis. We have amino acids that we use to make triglyceride to store as fat, or when we use amino acids to provide energy directly to the body. Remember I said that in any of those three instances, we need to get rid of the amine compound, the nitrogen. And I said that we're going to take that amine group, the NH2, and we're going to convert it to NH3, which is ammonia. And then I said we're going to convert the ammonia to urea, and then the kidney is going to process out all that extra urea in the urine, right? So this is the process of deamination. This is the process of getting rid of the amine group when we're going to use the carbon skeleton of the amino acid to make glucose, to make triglyceride, or to be used as energy directly. So do understand what deamination means. It's just the process of taking your amino acid. You're going to use the amino acid for glucose, a triglyceride, or energy or calories, right? And so in doing so, you need to get rid of the amine group. And you're going to convert the amine group to ammonia, NH3, and then to urea, and then excrete the urea. And this process happens at the liver. And this process happens at the kidney. Okay. Protein adequacy. Yeah, so this isn't really a term per se, but nitrogen balance is a good term to remember. So, well, even in practice, we don't really talk a lot about nitrogen balance. But nitrogen balance is basically, are you getting enough, right? Is the person eating enough protein? So we have positive nitrogen balance and negative nitrogen balance. And so this is, again, looking at the nitrogen. Let me go back here. The nitrogen, right, that is in the amine group, right? So if we're talking about nitrogen balance, we're basically talking about protein adequacy, because protein is the macronutrient that provides nitrogen. So if we have positive nitrogen balance, that means we're consuming more nitrogen than we are losing, right? Which is, we're going to see positive nitrogen balance at a few specific instances in life. Whenever a body is growing, so children, right, infants, children, adolescents, they should be in positive nitrogen balance, because they're building. So any extra protein that comes into their body, any extra nitrogen that comes into their body is going to get used to build new cells, build their bones, build their tissues, build their muscles, right? So anytime we see growth, pregnancy is also a time where we're going to see growth, right? We're seeing growth of the fetus. So that's also a time of positive nitrogen balance, where more nitrogen is coming in and being utilized than is being excreted as urea. We'll also see it when we're recovering from an illness or recovering from a protein deficiency. So as we're recovering, that process, like after you've been sick for a while and you haven't eaten anything at all, and then finally you start eating again, your body's recovering from illness. you're starting to consume protein-rich foods again, you're starting to consume nitrogen again, and your body's using it to build up new cells and tissues. So positive nitrogen balance. And then we have negative nitrogen balance when just the opposite, when we excrete more nitrogen than we consume. So really, we only see this during excessive low-calorie diets and during starvation. And then if we were to have like just nitrogen balance, that's where intake equals excretion. That's like, daily life once you're an adult and not pregnant or sick. So here are the images. So nitrogen, positive nitrogen balance. Remember that's during periods of growth, right? Growth, pregnancy, recovery from illness. And that should make sense too. Again, that shouldn't be something you have to memorize. But just once you understand what we need proteins for, you'll know that positive nitrogen balance is when we're growing, basically. And then you would know that negative nitrogen balance is when we're not taking in enough. So again, that can be during starvation. That could also be during severe illness, again, infection, burn, or injury, or on a very low-calorie diet. And then nitrogen balance is found in healthy adults who are not pregnant, right? So you're taking in just as much as you're going to need. That's nitrogen balance, a way of assessing protein adequacy. Alrighty, so now let's talk about how much for protein, right? So remember the term AMDR, acceptable macronutrient distribution range, and you should well, I do want you to know for this course, just to be able to know off the top of your head, what is the AMDR for protein, what is the AMDR for fat, and what is the AMDR for carbohydrate. I think I said that right. So for protein, it's 10 to 35% of total calories, right? So that's a pretty big range, but 10 to 35% of total calories should come from protein. Of course, it's a large range because there are times in life where we need more protein than others, right? Protein needs are higher during growth and pregnancy, just like we saw here. Protein needs are higher during positive nitrogen balance. So during these periods, during childhood, adolescence, pregnancy, lactation, also not just pregnancy, but even in the 1 to 2 years after when a female might be lactating, protein intake should be at this higher end range. So 25, 30, 35% of total calories. Also, people who are very active, so like elite athletes in particular, and even older adults, because their protein digestion and protein synthesis may not be as efficient, there may actually be higher need for older adults and very active individuals. And then I wouldn't say there's unanimously a higher need for people following a vegetarian or vegan diet. You just need to make sure you're getting your complete proteins, right? So eating soy, eating quinoa, but also doing your due diligence to combine proteins as needed to make sure you're not missing any of the essential amino acids. There is also an RDA for protein. We saw an RDA for carbohydrate too, which was 130 grams per day. For protein, it's based on body weight. And again, that's because, well, carbohydrate should be based on body weight too, but it isn't. So with protein, it's based on body weight because every body is different. So the basic RDA for sedentary people is 0.8 grams per kilogram of body weight per day, right? So kilograms, you can take your pounds. Excuse me, I'm gonna sneeze. To convert pounds to kilograms, you can just divide your pounds by 2.2, and that will give you your kilograms. So say we have a 70 kilogram person. Pull out my calculator here. So you would just take 70, multiply it by 0.8, and that would be 56. So for a 70 kilogram person, which would be roughly like 140, 150 pound, we need 56 grams of protein per day. Great. That would be at the minimum if they're sedentary. So as soon as they start to be more active than sedentary, that protein requirement would go up. So I think maybe I haven't written it in here, but RDA can go up to about 1.6. Well, it can even go as high as 2, but generally 1.6 is considered the upper end range at which intake above that level provides no extra benefit. So usually up to 1.6 grams per kilogram body weight. So again, that would be for like super active people. You may sometimes see a recommendation of 1.8 grams or even 2 grams per kilogram body weight, but my personal recommendation is 1.6 as an upper level. You can also always just go by the AMDR and see how your RDA compares to your AMDR, and make sure that you're keeping your AMDR within that 35% of total calorie range. So RDA, we could say, is on a range of 0.8 grams per kilogram body weight per day to really up to 2, but I'm going to write it as 1.6. And that's per day. Okay, so high protein intake, bone health. Oh, yeah. There is some question that higher intakes of animal, that high protein diets can increase calcium excretion, right? Not shown to lead to bone loss. Too little protein causes bone loss. So there has been some look at this, right? Because all that extra ammonia, all that extra, those extra amine compounds, the ammonia, the urea, that it could cause the body to become too acidic. And so we might need to leach calcium out of the bones to balance the acidity of the body. That's really kind of speculation at this point. There's some research suggesting maybe that's the case, but I would say at this point we don't really have enough information to prove that high protein diets can cause bone loss. And in fact, for sure, too little protein can definitely lead to bone loss. And especially in middle-aged and older women, it's really important to get enough protein, whether it be from animal or plant sources, because we know actually that adequate protein intake as we age is beneficial to bone health. Higher protein intake may be beneficial in healthy populations. Yeah. So there's some speculation that too much protein can be deleterious to our bones. I think we've yet to determine whether or not that's true, but definitely getting enough is critical without question. Kidney disease. So I did say earlier that too much protein can increase risk of kidney disease and also liver disease, especially in people who might already have other conditions that are affecting the kidney or the liver. So again, as much as 2 grams of protein per kilogram body weight per day is suggested to be safe for healthy people. So anybody who doesn't already have underlying risk or underlying kidney or liver issues. It is important to drink enough water as we increase our protein intake to help with that flushing that urea from the kidneys, right? Otherwise, it could lead to kidney stones. And then protein associated with elevated blood cholesterol levels. So what we do see is that high animal protein diets, because of the saturated fat, and the cholesterol, that can lead to elevated blood cholesterol levels. And we see, interestingly, that people who follow more of a plant protein diet have, like, plant proteins have very cardioprotective effects. So we can see elevated blood cholesterol from animal protein, from too much animal protein intake due to the saturated fat and the cholesterol, and that increases the risk for cardiovascular disease. So the recommendation is to limit saturated fat, to switch to plant proteins because you get fiber and you remove saturated fat and cholesterol, and to increase more of those healthy omega-3 fats. Where do we find protein in our diet? So I think the obvious, I said meat, dairy products, and eggs. And then our plant foods would be all beans and lentils, which we call legumes, or whole grains. Remember when we talked about the bran, the germ, and the endosperm? We saw that the germ and the endosperm are protein rich, so long as they're not processed. So whole grains are a great source of protein. Nuts and seeds are a good source of protein. And vegetables you saw can give us some protein as well. There are now, in the 21st century, we've got lots of made up foods like corn or the Impossible Burger or the Beyond Meat. So there's a lot of stuff where we're kind of like making protein foods. Those are not really the healthiest for us. I don't know why ancient grains goes here. Ancient grains are really just whole grains. But these are just grains that have been around for a long time, but just haven't been mass produced. So arguably they might even be healthier for us because they should have pretty high protein content and pretty high fiber content. These would be helpful charts if you're interested. Again, this could be another thing you do for your My Diet analysis is to check to make sure you're getting adequate protein. Even consider including some plant sources of protein to reduce your saturated fat and cholesterol intake. So you've got beef, poultry, seafood here, pork, dairy, and then down here you have soy products. You'll see that, also you'll see the serving sizes, 3 ounces. So that would be something you could do too. So check to see how much you're eating, right? Like a 1/4 pound burger is 8 ounces. That's almost 3 servings. And 3 ounces already gives you 22 or 27, or 29 grams of protein. So depending on your body, your kilograms, your body mass, you may already be well on your way to your protein intake for the day. And then you'll see 3 ounces of tofu is going to give you significantly less protein, but still a good amount. Tempeh, this is another fermented form of soybeans, or you can make tempeh from grains as well. You'll see that can be a really rich source of protein, just a 1/2 cup. And then we've got beans, like we said, and nuts are other good sources of protein. So again, a 1/2 cup is going to give you a bit. Again, also going to give you some fiber, which is excellent, and no saturated fat. That would be a cool chart if we could see the amount of saturated fat relative to protein, relative to fiber. Okay, so a little bit on vegetarian and plant-based diets. So in vegetarian, you just eat mostly vegetables. I don't know if we have it in here. Yeah, there's all these different names for different types of vegetarian diets, like semi-vegetarian or flexitarian, where they eat primarily vegetables, but may also consume seafood, poultry, eggs, or dairy. Lacto-ovo is probably the most common one. So people eat vegetables, grain, all plant foods, plus lacto, meaning dairy products, and ovo, meaning eggs, but no fish, no meat. Pesco-vegetarian would be, eats vegetable foods plus fish. Pes means fish, so they might consume fish in the diet. Then vegan is basically only plant foods, so no animal foods whatsoever. Macrobiotic is like a type of vegan diet that's like really only eating super healthy plant foods, so like eating plant foods that aren't processed. So I don't know, they say it can cause malnutrition and death. That's not untrue, but that's, again, if it's taken to the extreme. But macrobiotic is probably one of the healthiest ways we can eat. I think it's just can be more challenging in the 21st century because it's hard to access such pure and clean food anymore, sad to say. And then fruitarian is very interesting. I have known some people personally who follow fruitarian diet, and they do well. Can be worth supplementing if someone's following a fruitarian diet, because particularly like B12, iron, zinc, and riboflavin can become pretty low on a fruitarian diet. But it's where they literally really only eat the fruit portion of the plant. So people choose vegetarianism for a variety of different reasons. In general, plant-based diets are the healthiest way to go. But that doesn't mean that we have to eat only plants. I say plant-based intentionally, right? Even a plant-based diet that allows for fish sometimes, or a plant-based diet that allows for eggs periodically, that's much healthier than, let's say, a hamburger and hot dog diet. People might also choose vegetarian for ecological reasons. We're starting to have a lot of scientific research now showing the environmental impacts of plant agriculture relative to animal agriculture, and plant agriculture has unanimously lower impacts on the environment relative to growing livestock or growing animals for meat. Oftentimes there are religious reasons why someone might follow a vegetarian diet, also ethical reasons, like for a lot of folks, we now live in an age where we have enough food to eat and eat very well without killing or harming other animals. So a lot of people choose a vegetarian or vegan diet to do less harm, and then also we'll talk about in Unit 4, food safety issues. Most foodborne illnesses are through meat or dairy products. So following a plant-based diet, again, not necessarily 100% plant-based, but eating more plants than animal, reduces the risk for food-borne illnesses. And then again, eating more plants reduces the risk for cardiovascular disease, reduces the risk for diabetes, reduces the risk for gastrointestinal disorders. The health benefits of plant-based eating are vast. So whole food plant-based, this has been pretty trendy lately. So whole food plant-based eats primarily plant foods that are in their whole, hence whole food, original form with minimal to no processing. This is almost like modern day macrobiotic. Yeah, almost. But anyway, this is also a very, very healthy way of eating. It's basically like eating vegetarian or vegan as healthy as you can be. So it's distinct from vegetarian and vegan in that no processed foods are eaten, whereas Oreos are vegan, so you could have a vegan diet that's all Oreos, so it's not very healthy. So the food groups again would just be all of your plant foods, so fruits, vegetables, beans and lentils, nuts, seeds, whole grains, water, of course. There's been really cool research showing that a whole food plant-based diet can reverse cardiovascular disease and type 2 diabetes. Not just manage well, but quite literally reverse these diseases. It's really, really, really fascinating. You might look up Dean Ornish if you're interested. Dr. Dean Ornish, he's one of a few doctors who did a lot of research here, but fascinating what he's done with a lot of his patients and whole food plant-based way of eating. Okay, so what are the health benefits, right? So you're gonna have far less saturated fat, far less cholesterol. Usually we take in less energy too because there's so much more fiber. So we wind up having a lower calorie diet when we're eating plant-based. Blood pressure is also going to be lower because it's usually a lower sodium way of eating. Again, less saturated fat, usually less sugar, less cholesterol. So fewer heart issues, fewer digestive issues, can reduce the risk for some cancers as well because, as we'll see in Unit 3, plant-based diet is really rich in anti-inflammatory and antioxidant compounds. And we can also see reduced risk for kidney disease, kidney stones, and gallstones, again, because there would be less of that urea, that deamination process making urea. Okay, but some dietary considerations for whole food plant-based. So remember the complementary proteins. That's really important. At the very least, including quinoa, but because these are complete proteins. But then otherwise make sure to do that protein combining. So that chart that we looked at earlier to help figure out complementary proteins, something like that I think is an absolutely invaluable resource to somebody planning a whole food plant-based way of eating. You may also consider supplementing with vitamin B12. Anybody could supplement with vitamin D because that's one we make in our skin. And so in the wintertime in northern latitudes, we all become vitamin D deficient. We'll talk more about that in Unit 3. B2, and then iron, calcium, and zinc. In particular, in my experience, B12 and zinc, it just depends on the way the person likes to eat. We'll go through each of these minerals later on. Iron is quite abundant in beans and lentils. So as long as someone's getting enough beans and lentils, and then we also have it in many of our other foods, like even chocolate is a good source of iron. So including some cocoa powder or cocoa nibs in various recipes can be another little iron addition. A lot of our plant foods are really rich in calcium as well. So again, it's just a matter of eating enough, but good to stay, to stay educated and stay aware and make sure that you're not starting to slip in any of these nutrient levels. Again, the My Plate and the Harvard Healthy Eating Plate that we looked at in Unit 1 can be really, really helpful resources for planning a vegetarian, vegan, or whole food plant-based diet. Okay, so then this is going to be the last section for this lecture. So we'll talk about protein, energy, malnutrition, and then we'll end with a quick slide on disorders or diseases associated with protein synthesis. So protein, energy, malnutrition, again, malnutrition meaning poor nutrition. So in this case, we're talking about inadequate or undernutrition. So inadequate protein intake. There are two forms, marasmus and kwashiorkor, and we see both of these primarily in developing countries where total energy intake is low. So protein energy malnutrition is a disorder caused by inadequate protein intake and inadequate energy intake overall. So usually we only see protein energy malnutrition where we also see severe lack of calories. So marasmus. This is basically brought about by just overall not enough calories, not enough food for the person. So again, we see this more so in developing countries. What happens then? We definitely see muscle wasting and just overall weakening of the body. We're going to see stunted growth, stunted cognitive development. We would see low body temp, slow metabolism, stunted physical growth, and basically a wasting body, right? Severely weakened immune system. You're going to see low red blood cell levels, anemia. Certainly going to see fluid and electrolyte imbalances. So marasmus. Oh, this is an image of marasmus here, right? So you can see just kind of wasting, sadly, right? This young child is definitely not getting enough nutrition. You can see the rib cage quite clearly. You could even kind of see that the neck is super thin. We can't see much about the rest of the body, but you can see it's just not getting enough nutrition to really actually grow this little body. And then kwashiorkor is interesting. Again, a protein deficiency. So where we see kwashiorkor most often is when an infant is not breastfed long enough and they're transferred to like a solid food or kind of a cereal diet that isn't providing enough protein. So again, we're going to see similar issues here. So we're going to see weight loss and muscle wasting, or retarded growth and development. We'll see fatty degeneration of the liver, sores and skin problems, hair loss, and then even, again, like cognitive or emotional decline. So in kwashiorkor, that's where we see this edema of the stomach, right? So we're not having enough proteins to pull some of that fluid back out, not the stomach, but the gastrointestinal region, so the abdomen. So you see tell signs of kwashiorkor is this abdomen. And then you can see, again, just kind of stunted growth. Like these look like pretty small arms for this child, short, even like short bones. Obviously, you can see the full rib cage. So again, overall, just not enough energy. And then also protein deficiency. Okay, and then I think this is the last slide. So some genetic disorders related to protein. So remember, proteins are coded by our DNA. So genetic disorders would affect protein synthesis. So 3 examples, we have phenylketonuria. And we mentioned this before when we were talking about essential and non-essential amino acids and the instance in which we might have conditionally essential amino acids. So in phenylketonuria, the person is unable to like fully break down phenylalanine, one of our amino acids. So there can be a lot of like metabolic byproduct buildup that can lead to permanent brain damage. In sickle cell disease, what we see is improper formation of the red blood cells. So the red blood cells form in this sickle shape. And so that can lead to poor blood flow, can actually lead to blockage of the blood vessels, and that can damage organs that might not get enough blood supply. And then lastly, we see something called cystic fibrosis. Again, so all of these, phenylalanine being an amino acid, hemoglobin being a protein, right? And in cystic fibrosis, again, we see an abnormal protein synthesis that then affects the way that chloride gets transported in the body, and that can also lead to basically organ dysfunction. So what happens in this case with cystic fibrosis is that certain organs in the body that produce mucus start to produce a really, really thick, sticky mucus. And so that can start to like clog and back up some of these organs. So we can see pretty intense respiratory distress. That's one system where we see really important mucus production, and also the gastrointestinal system. So you can see issues of the GI and respiratory system. Those two are most affected by cystic fibrosis. So 3 examples of genetic disorders. Again, genetic disorders meaning poorly produced proteins, basically. And then here's just a visual representation of a sickle cell. So you can see these are normal red blood cells. They have that nice kind of round, fluffy shape. And in the sickle cell, it just didn't form properly because of the genetic mutation. So again, these will get stuck and lodged in the blood vessels, leading to organ damage when enough blood is not supplied to the organ. Okay, so that's the end of the protein lecture. It's a little bit of a shorter one. So if you do have any questions, email me as always. Hopefully you have a little extra time this week, so please do be working on your My Diet Analysis project and making sure you're up to speed on all of the quizzes. Alrighty, thanks everybody, and I look forward to seeing you next time for Chapter 7.